Well, welcome to the 22nd annual Lefeber Winter Series on Aging in honor of Dr. Lefeber. For those of you in the audience who don't know who I am, my name is Blake Rasmussen. I'm the chair of the nutrition department here at UTMB and good friends with our speaker today, so I'm introducing him. So, I'd like to give you a little bit of a brief introduction about who Dr. Adams is. So Dr. Christopher Adams is a professor of medicine at the University of Iowa Medical School. He's an endocrinologist as well, and he's doing basic science research into the mechanisms of why older folks lose muscle and maybe what are some potential therapies that we can use as we age to stop muscle loss and maybe even grow our muscles somewhat. So a little bit of his background. He has a bachelor's degree in cell biology from the University of Kansas. One thing that I found fascinating was that he actually, as an undergraduate student at Kansas, got a paper in the Journal of Cell Biology as an undergraduate student. I found that very cool. He then was accepted into the University of Iowa MD PhD program, where he did his medical school training and did a PhD in molecular physiology and biophysics. He then moved to the great state of Texas to do his residency and fellowship training in Dallas at the University of Texas Southwestern. And after his residency and fellowship, he thought he would join a lab that helped learn. He wanted to do science he wanted to be a physician scientist, having done the MD PhD program, and he joined the lab of Brown and Goldstein. For those of you who may not be familiar with those two guys, they won the Nobel Prize in 1985 for studies in cholesterol metabolism. And then he also, his PhD advisor was another member of the National Academy of Sciences. So his training was by 3 National Academy of Science people, so it was very impressive. A few other things. He is the founder and CEO of a startup biotechnology company called Emion. And he'll show you some slides today about what his company is doing in an effort to counteract muscle loss with aging. So I will turn the time over to Chris and welcome to Galveston.
Well, thank you very much for that kind introduction, Blake. It's a real pleasure and honor to be here today, be back in Texas, so it's great. So I thought today, the title that I think you may have seen on the billboard was about age-related muscle weakness and atrophy, but I thought today we'd just talk about an aspect of the work that we're doing and really just how we're trying to search for ways to reduce this problem in people. Yesterday I gave a different talk to the basic science group talking about mechanistic work that we're doing. I thought today we'd talk about ways that we're doing the best we can to try and find ways to reduce muscle wasting during aging. So first of all, disclosures, my funding sources, equity. So the major focus of our laboratory is muscle wasting. Another word for muscle wasting, another thing that's called is skeletal muscle atrophy. So muscle atrophy has many different causes, including not just aging, but also muscle disuse. So for example, when people wear a cast or are subjected to bed rest, or have a stroke or a spinal cord injury. Critical illness, so when people are very sick in the intensive care unit. A variety of chronic illnesses. Almost every chronic illness can lead to this condition of muscle atrophy, including diabetes, renal failure, heart failure, cancer, lung disease, liver failure, and chronic illnesses like HIV. And then finally, malnutrition or starvation can cause our muscles to undergo wasting. So this is a very, very common problem. A lot of different ways, a lot of different causes. In addition to being very common, this can have very serious effects for people. So for example, it can lead to a loss of strength and endurance. It can delay the recovery from an illness or an injury. It impairs mobility. It actually increases mortality or increases the risk of death in people with chronic diseases and in the elderly. And of course, it can lead to a loss of independent living or nursing home placement, which is something we all want to avoid. So all of these things place very large burdens on patients and their families and really society in general. But although this condition is very common and serious, currently we don't have any reliably effective ways to prevent or treat it. And this graph illustrates the scope of the problem in the United States. So what we have here is the treatment availability versus the prevalence in the United States. And we're looking at 5 major medical problems. And as you can see, muscle atrophy, it's almost as common as hypertension or high blood pressure. But we have 0 medicines for it. So what we are doing is in the laboratory is investigating how muscle atrophy occurs at the molecular level, the biochemical level. And then we're trying to use that information to find chemical compounds, or another word for that is small molecules, that we might be able to someday use to prevent or treat muscle atrophy in people. And the way that we look for small molecules is really fundamentally different than a traditional drug discovery approach. So typically when people want to discover a drug, they start with a very well-defined molecular target, something like an enzyme or a cell surface receptor. But we can't really do that with muscle atrophy because the condition, it's very complicated at the molecular level, and it's also grossly understudied. So we don't have a lot of well-defined molecular targets. So we took a different strategy. And I'll try and go through it, and if anybody has any questions, please stop me. But basically, even though we don't know a lot at this point about how muscle atrophy occurs at the molecular level, one thing we do know is that it involves widespread changes in skeletal muscle gene expression. So when a stimulus for muscle atrophy, so for example, fasting or muscle disuse or aging or illness, when this is superimposed on a muscle, there are hundreds of changes in muscle gene expression. So many genes get turned on and many genes get turned off. And we know that at least some of those changes in muscle gene expression are required for the muscle to waste away or undergo atrophy. So many genes get turned on, many genes get turned off, and this generates something that we call a molecular signature, and that's illustrated here by this slide. So you start with a muscle atrophy stimulus, and that leads to activation of genes and inactivation of genes. When a gene gets activated or turned on, that gene then generates a specific chemical called an mRNA. That mRNA is specific for that type of gene. So when hundreds of genes get activated, hundreds of specific mRNAs increase in the muscle. And conversely, when the genes that are inactivated, those mRNAs decrease. Okay, and then the vast majority of genes are unaffected, so the levels of those mRNAs don't change. The total pattern of changes in these mRNAs, we call that an mRNA expression signature of muscle atrophy. And importantly, right now with the current technology that's available to us, we can do it in one experiment. We can determine, we can measure the level of every single mRNA in a muscle in one experiment, and so we can get this signature of muscle atrophy. So as we began to think about how to search for chemicals to reduce muscle atrophy, we hypothesized that chemical compounds with signatures that were like the exact opposite of muscle atrophy, then if we could find compounds like that, they might actually reduce muscle atrophy. So the idea is here. So take, for example, a signature of muscle atrophy. We know hundreds of mRNAs increase, hundreds decrease, and most of them don't change. We thought a hypothetical, or our theoretical chemical inhibitor of muscle atrophy, it would be the opposite of this. So these mRNAs, this chemical, instead of increasing these mRNAs, it would decrease these mRNAs. And instead of decreasing these mRNAs, it would increase these mRNAs, and it shouldn't affect these mRNAs. Okay. So another way to think about this would be if we could know the mRNA expression signature of muscle atrophy and then find small molecules whose signature negatively correlated to the signature of muscle atrophy, then maybe those small molecules might inhibit muscle atrophy. And so to test this hypothesis, we needed signatures of both small molecules and muscle atrophy. The small molecule signatures that we used came from something called the connectivity map. This is an open access project that was conducted at the Broad Institute. And what the investigators did is they treated human cell lines, so these are cultured cells, with chemical compounds or small molecules, and then 6 hours later they isolated the RNA from those cells and did experiments to determine the mRNA expression signature. The muscle atrophy signatures came from experiments we did in our laboratory. We studied humans and mice, and we obtained muscle biopsies, or pieces of muscle, from the humans, from the people, and from the mice under normal conditions, and also during conditions that cause muscle atrophy. And then we took those muscle samples, isolated the RNA, and then did experiments to determine the mRNA expression signatures. And from those data, we generated 2 different genome comprehensive mRNA expression signatures of skeletal muscle atrophy. The first one shown here. And this consists of mRNAs that are similarly affected by fasting or starvation in both human and mouse muscles. So this would be sort of an evolutionarily conserved picture of how fasting affects gene expression in skeletal muscle. So this was our first signature, and you can see there are a number of mRNAs that were increased and a number of mRNAs that were decreased. And in many cases, we don't even know what these mRNAs do. I mean, we know they encode for a protein. We don't know how that protein fits into the pathogenesis of muscle atrophy, but nonetheless, this is a signature, a pattern that we can use. The second signature that we generated is shown here, and this consists of mRNAs that are similarly affected by fasting and spinal cord injury in humans. So now we're looking at the conserved effects of 2 very different types of atrophy stimuli, starvation and spinal cord injury. And here you can see there's a smaller group of shared effects, but nonetheless, this is another signature. So we have these 2 signatures of muscle atrophy from humans and mice, and we took them and we compared them to the small molecule signatures from the connectivity map, looking for small molecules or chemical compounds whose signatures negatively correlated to both of the muscle atrophy signatures. And if they negatively correlated to both of them, then we considered that a very good candidate as a potential way to reduce muscle atrophy. And to test that, then we took those small molecules and we tested them in mouse models to see whether or not they would work. So through these studies, we identified 2 compounds that we've studied the most up to this point. And they're shown here. One's called ursolic acid and the other is called tomatidine. So these are both natural compounds. Ursolic acid they call it a pentacyclic triterpene acid. It's found in the peels of apples and a number of other types of edible fruits and herbs. Tomatidine is what they call a steroidal alkaloid, and it comes from tomatoes, particularly green or unripe tomatoes. So both of these compounds, they're found in apples and tomatoes. They have favorable safety profiles, and that's critical for what we're ultimately trying to do. The effects of these compounds on muscle were not known, but the finding that their signature negatively correlated to a signature of muscle atrophy, that suggested that they might inhibit muscle atrophy. And so to test that, we did a series of studies in young adult mice, and they're summarized here. So these are young adult mice, not old. And it turns out if you subject a mouse or a human to fasting or immobilization, then the animal undergoes muscle atrophy. We performed experiments and we found that if we administered either ursolic acid or tomatidine to the animals during the period of fasting or immobilization, they underwent less atrophy. So that told us that these compounds had a capacity to at least partially prevent acute muscle atrophy caused by starvation or by immobilization. We also performed experiments to see if they might enhance the recovery from muscle atrophy. And the way we did that is we used a model that we call limb immobilization. This is very similar to wearing a cast. So if you ever wore a cast or know someone who had to wear a cast, the muscle under the cast gets much smaller. Well, the same thing happens in mice, and we can model it like this. And the way we do that, we basically place a cast on one leg in the animal. Over the course of a week, that'll cause muscle wasting in that leg, localized only to that leg. The other leg remains mobile, and it serves as a control, so it actually doesn't undergo atrophy. So to test whether ursolic acid and tomatidine might enhance the recovery from atrophy, we did this experiment. So So here on this graph, we have time on the x-axis, and on the y-axis here we have muscle weight. We're comparing the weight in the left leg to the weight in the right leg. So for the first 7 days of the experiment, the left leg was immobilized, and that induced atrophy as expected. So we got about a 20% loss of muscle mass. At that point, we remobilized. We took off the cast. We remobilized the left leg and allowed it to recover. But during that recovery period, we administered either a control compound or ursolic acid or tomatidine to these mice. And what we saw is that the muscle came back faster if we administered these compounds. So that told us that they had a capacity to enhance the recovery of muscle mass after immobilization. Now, when you think about muscle mass and muscle atrophy, it's very important to look at the muscle fibers or the cells in the muscles. So when we have muscle wasting, we don't actually lose cells in the muscle. The number of cells stays the same, but the cells become smaller. And so to really determine whether these were affecting muscle atrophy, you have to look at the cells, and that's shown here. This shows cell size or muscle fiber diameter. We call them muscle cell and muscle fiber. So this shows fiber diameter, and this shows the percent of fibers, percent of total fibers. So you can see that we have 3 conditions, and in each condition there's roughly a bell-shaped curve. But the curve is shifted to the right with ursolic acid and tomatidine. That means the fibers are bigger or they're less atrophic, and this confirms that they enhance the recovery from muscle atrophy. So these studies in acute models of muscle atrophy really set the stage for studies of the condition that we're most interested in. I think most of the people in the room are most interested in, and that's aging. So age-related muscle weakness and atrophy, this is a major public health problem. It affects virtually everyone. At around age 30, even elite athletes begin to lose strength and muscle mass. Over the next several decades, the rate of strength loss is 3 times the rate of muscle loss. So, in other words, strength per unit muscle goes down. They call that muscle quality. So the reduction in strength per unit muscle mass or muscle quality, that's really a hallmark of the aging process. By the time people reach 65 years old, almost everyone has experienced a significant loss of muscle strength, quality, and mass. And I think almost everyone would say that they're not as strong and they're not as muscular as they were when they were, say, 25 years old. Now, the molecular basis, how this occurs at the molecular level is very poorly understood at this point. It's a difficult condition to study because it takes a long time to develop. And of course, like the other types of muscle atrophy, we don't have good ways right now to prevent or reverse the loss of strength and muscle mass during aging. So fortunately, we can model this condition in mice. Mice are considered they develop a lot of the same features of muscle aging that people do. And in this graph, what I'm going to show you is some data of how we study this condition in mice. So here what we have are mice that are 6 and 22 months old. So a 6-month-old mouse is like a middle-aged mouse. A 22-month-old mouse is elderly. So these mice, they're matched for their total body weight. And here what we're looking at in this graph is the skeletal muscle weight. We're looking at the combined weight of the quadriceps and triceps. Those are the largest muscle in the hind limb and forelimb. And I'm going to show several graphs like this. And in these graphs, each symbol represents one animal, and the horizontal bars are the mean and the standard error. So these mice, these 2 groups of mice, they differ in age. They have the same total body weight, but the amount of muscle is significantly reduced in the elderly mice. It's reduced by 18%. This loss of muscle mass with aging is explained by a reduction in the size of the muscle fibers, the muscle cells. Okay, so here, these pictures here are cross-sections through muscle. Each of these shapes is one muscle fiber. These are real long cells, but we're taking a cross-section through them. And you can see the muscle cells in the older mice are much smaller. The data are, we can quantitate these data here. For example, here's the fiber diameter, average fiber diameter in each muscle. It's reduced by about 14%. If we look at the fiber size distribution curve, the curve is shifted now to the left, so the fibers are smaller. So 22-month-old mice have an age-related muscle wasting. They also have age-related muscle weakness. So we have two ways to look at that. One way is we do a test called in vivo grip strength. And here we see that there's a 12% reduction in strength in the older animals. And then an even better test is shown here. We call this ex vivo specific force. And so here what we're doing is we're actually taking a muscle out of the animal. We're stimulating it with electrodes and measuring the force generation. And then we're normalizing the force to the size of the muscle. So this is really muscle quality or strength per unit muscle mass. And as you can see, that's reduced by 36% between 6 and 22 months. So together what these data tell us are that 22-month-old mice have significant age-related muscle weakness and atrophy. And this is consistent with what many other people have reported, and it's very similar to what we see in elderly people. So from this perspective, we asked whether ursolic acid and tomatidine might have a capacity to reduce age-related muscle weakness and atrophy. And to test that, we got a grant from the National Institute on Aging. They gave us some elderly mice that were 22 months old, and we gave those mice, we gave them food, either regular mouse food or food that had been supplemented with either ursolic acid or tomatidine. So we just incorporated these compounds into the food. So we let the mice have free access to these diets for 2 months. And altogether in these experiments, we had 32 mice in each group. And at the end of 2 months, 4 of the control mice died of natural causes, which is really consistent with their advanced age. But only 1 of the ursolic acid mice died, and none of the tomatidine mice died. So we developed an assay where we could measure the level of these compounds in the mouse bloodstream, and that's shown here. So this shows the plasma level at the end of 2 months. So we have some mice that are on the control diet, and we're looking at the amount of ursolic acid and tomatidine in their blood. And you can see if they ate a normal mouse diet, there was no ursolic acid or tomatidine in the blood. If they ate the ursolic acid diet, they had a higher level of ursolic acid in their bloodstream. And if they ate the tomatidine diet, they had a higher level of tomatidine. So then we looked at the muscles. So this shows muscle weight. Again, we're looking at the quadriceps and triceps all combined. And as you can see, there was a significant increase with both ursolic acid and tomatidine. There was a 9% increase with ursolic acid and a 10% increase with tomatidine. The increase in the muscle weight was explained by an increase in the size of the muscle cells or the muscle fibers. So these are representative images from the quadriceps muscle. You can see the fibers are larger. These are the quantitation of the data, the muscle fiber size distribution curves. You can see both compounds shift the curve to the right. So they reduce muscle atrophy or muscle wasting. But the thing we really care about is function. And so we looked at strength. So this shows grip strength. We saw a significant increase with both ursolic acid and tomatidine. There was a 12% increase with ursolic acid, a 10% increase with tomatidine. And then we looked at specific force. That was the measurement of muscle quality or strength per unit muscle mass. And we saw a 30% increase with ursolic acid and a 33% increase with tomatidine. So together, what these data indicate is that ursolic acid and tomatidine, if we supplement the diet of elderly mice with these compounds, we see a significant reduction in age-related muscle weakness and atrophy. So there's an increase in muscle strength, mass, and quality. And furthermore, I think we can say the magnitude of the changes suggests that the compounds are largely restoring these parameters to middle-aged adult levels. So we're excited about the potential, translational implications of this. Maybe we could use this information to find ways to help people, maintain muscle mass during aging. We're actually, Blake and I, Dr. Rasmussen and I are starting a project right now to explore that. So we're excited about the translational implications. At the same time, we thought that we might be able to use these two compounds as tools to better understand how a muscle undergoes aging. Why does muscle become weaker and more atrophic with aging? So we use them as tools to investigate the pathogenesis. And I don't have a lot of time. I don't think I have time to go into this whole story in detail, but I think it's turning into an interesting story. So what we found is that through those studies, we identified a protein called ATF4, and it turns out that this protein Here's a picture of the protein. Its molecular structure. It's a transcription factor. This is a protein that turns on genes. We found that ATF4 is a key protein target of ursolic acid and tomatidine in aged muscle. So it turns out by studying those muscles from those mice that ate the diets containing either the control diet or ursolic acid diet or tomatidine, we found that in those muscles, ursolic acid and tomatidine reduced ATF4 activity. Okay, so made this protein less active. So that suggested that ATF4 could be a mediator of muscle aging, a cause of muscle aging. And to test that, we generated and studied some genetically modified mice that lack ATF4 in their muscle fibers. And what we found is that if we eliminate the ATF4 gene in muscle fibers, that prevents age-related muscle weakness and atrophy, very similar to ursolic acid and tomatidine. So we think that this is important because it begins to explain how these compounds are reducing muscle weakness and atrophy during aging. We also think it's important because this is now the first example of a protein that's responsible for the loss of muscle mass and strength and quality during aging. And so now it gives us a biochemical handle on this process. Now we have a way where we can study what regulates that protein, how does that protein cause atrophy, and I think it gives us a way now we can really get some deeper insight into the causes of muscle aging. And that's another thing that we're working on with some of our colleagues here at UTMB. So together, those data told us that ursolic acid and tomatidine have a capacity to reduce muscle atrophy during both acute and chronic conditions. And as a result of those effects, that increases strength and specific force or muscle quality and muscle mass. Our next question was whether ursolic acid and tomatidine might stimulate something called muscle hypertrophy. Okay, so here we have three muscles, pictures of three muscles. So here in the middle, this would be, say, a basal state of a muscle. From this basal normal state, a muscle can undergo atrophy and become smaller, or it could go the other direction. It could undergo hypertrophy and become bigger. Okay, so this is another word for this is muscle growth. So we wondered, since ursolic acid and tomatidine could take the muscle from here to here, we wondered, could it take it from here to here? So as one way to test that, we performed a series of studies in cultured muscle cells. Okay, so these are now, we call these cultured skeletal muscle tubes. They're cultured skeletal myotubes. They're basically muscle cells in a petri dish, okay? And we can grow these, we can isolate them from people or from mice and then grow them in a dish. And what we did a series of experiments. We found that if we added either ursolic acid or tomatidine directly into the dish, then that caused these cells to make more protein and become larger. So they stimulated hypertrophy. And this is, I think, an important experiment for us because what it tells us is if you eat a compound and you see an effect in muscle, you don't know if it's a direct effect on muscle. It could be an effect on the brain, pancreas, whatever. But this experiment begins to tell us that these two compounds act directly on muscle cells. Also, it tells us that they have a capacity to act on muscle cells from either humans or mice. And that means that humans and mice have the receptors for these compounds. So an example of one of these experiments is shown here. So here what we did was we took myotubes and we treated them with either ursolic acid, and this is at a concentration of 5 micromolar, or tomatidine at a concentration of 1 micromolar. And we treated with the compounds in the absence and presence of another compound called rapamycin. Rapamycin is a chemical that inhibits a protein that we know is very important for muscle growth. It's a protein that Dr. Rasmussen studied extensively called mTOR complex 1. So we know that rapamycin inhibits muscle hypertrophy. And so we did this experiment. We looked at ursolic acid and tomatidine with and without rapamycin. So here, this shows the average myotube diameter, and this is after 48 hours of treatment. And you can see the compounds increase the size of the muscle fibers. And here's another way to look at the data. Here, myotube diameter, percent of myotubes, you got the vehicle, ursolic acid and tomatidine. You can see these curves are shifted to the right. However, rapamycin blocks this effect. So you can see there's no effect in the presence of rapamycin, and all the curves sort of collapse to the same point. So what this tells us is that these compounds have a capacity to stimulate this hypertrophy pathway mediated by this mTOR complex 1, at least in myotubes. But what about in a real animal, in vivo? So to test that, we did experiments like this. So for example, in this experiment, we took young mice, young growing mice, and we gave them mouse food or mouse food that was supplemented with ursolic acid. And the mice consumed these diets for 5 weeks, and then we looked at the muscles. So this shows quadriceps weight. And again, each symbol is one animal. And you can see that the quadriceps weight increased by 18%. We saw a similar increase in all the other muscle groups that we looked at. And this was explained by an increase in the size of the muscle fibers. And it was associated with increased strength and muscle quality and endurance exercise capacity. We did similar experiments with tomatidine. So here we took 7-week-old mice. We treated them with either standard chow or standard chow that was supplemented with tomatidine. Mice consumed these diets for 5 weeks, and then we looked at the muscles. So here we have muscle weight. We're looking at 4 different muscle groups: the tibialis anterior, the gastroc, which is in the calf, the quadriceps. This is a thigh muscle, and then a triceps muscle that's up here. So as you can see, in each muscle, there was a significant increase in muscle mass in the mice if they were supplemented with tomatidine. The overall increase in 5 weeks was 14% increase in muscle. And the increase in muscle mass was explained by an increase in the size of the muscle fibers. And importantly, it was associated with improved muscle function. So grip strength was higher, specific force or muscle quality was increased, and running distance on a treadmill was also increased. So together, these data told us that these 2 compounds have a capacity to take a muscle from atrophic to here, but also to stimulate hypertrophy. So as we were doing these experiments, we saw something that was interesting to us, and that was, muscle makes up a large percentage of our total body weight. But so if you had a 14% increase in your muscle mass, then you would expect that your overall body weight would increase. But what we found is that the overall body weight did not increase, and that meant that another tissue must have decreased in weight. Okay, so the increase in muscle mass must have been offset by a loss of some other tissue. And it turns out that the tissue that was reduced in size was fat. Okay, so these are pictures of muscle fibers and fat cells from mice that were supplemented. Their diet was supplemented with ursolic acid. And you can see the muscle cells became larger, but the fat cells became smaller. So here's another piece of data from a similar experiment. So here we had young mice. We treated them with, we gave them either standard mouse food or mouse food that was supplemented with ursolic acid for 7 weeks. And this shows the final body weight. So they start at the same body, they're all matched for the same body weight at the beginning of this experiment. At the end of the experiment, you can see final body weight, there's no change. However, the muscle weight is increased by 14%. The fat pad weight, so fat deposits in their body, that was decreased by 35%. And the ratio of muscle to fat was increased by 66%. So when we saw this reduction in fat, we started to think about other metabolic parameters. So for example, one thing we looked at was fasting blood glucose. So we asked whether ursolic acid might decrease fasting blood glucose, and when we did this, we compared ursolic acid to a drug called metformin that we give very commonly to people with type 2 diabetes. And we found that ursolic acid reduced fasting blood glucose. Actually, it reduced it even a little bit more than metformin, and the dose was actually less. It required less ursolic acid to reduce blood glucose than the dose of metformin. So the finding that it's reducing fat and blood glucose, that led us to then start thinking about obesity. So to do obesity experiments with mice, we changed to a totally different diet. We moved to something called a high-fat diet. And this diet, it actually looks like cookie dough. It's sort of like mouse junk food, and mice love it, and they eat they eat it, and when they eat it, and they rapidly develop all the expected problems. So obesity, prediabetes, and fatty liver disease. So we said, what would happen if we put a little bit of ursolic acid into this high-fat food? So now we got two groups of mice on a high-fat food or high-fat food that was supplemented with ursolic acid. And this shows body weight on the diet. It was a 6-week experiment. Now these are young mice, so they're still growing. And so the body weight keeps going up in both of them, but you can see the mice that were treated with ursolic acid, they gained less weight. So there was some protection against the development of obesity in these high-fat fed mice. So then we thought about diabetes. Okay, so in people, we say a normal blood glucose is below 100. If it's above 126, we call that diabetes, and if it's between 100 and 126, we call that prediabetes. So what this graph shows is fasting blood glucose, and here's the dashed line is 100. So anything below 100 would be normal. So we have two groups, again, high-fat food supplemented with ursolic acid. The mice that ate the regular high-fat food, you can see all but one of the mice developed either diabetes or prediabetes. But ursolic acid prevented that, so it prevented prediabetes. Then the next thing we looked at was fatty liver disease. So fatty liver disease, they call this non-alcoholic fatty liver disease, or there are a number of different terms for it, but this is something that's becoming a very large problem, especially in the United States. It's actually beginning to eclipse alcohol as the leading cause of liver failure in the United States. So it's related to obesity, and it involves the deposition of fat into the liver. At this point, we don't have any good medicines for this problem of fatty liver disease, but we thought, anyways, in these mice, we took a look at the livers. So here we have sections through the livers without ursolic acid and with ursolic acid. So these are pictures under the microscope, and what we've done with these sections, we've stained the sections with osmium, and osmium is a chemical that it turns fat a black color. And you can see these livers are full of fat, but these livers are not. Okay, and these mice had elevated liver function tests. Okay, so AST, ALT, things like this, elevated, normal. If we looked at the liver weight, which is shown here, normal liver weight's 1,500. These mice that ate the high-fat food without ursolic acid, they all had large livers because they were full of fat. But these livers were normal size. So through these studies, we could see that ursolic acid and tomatidine were reducing obesity and some of its most serious complications. So how could this be? So from the simplest perspective, how could you lose weight or reduce obesity? There are two ways really. One is by reducing food intake. So maybe ursolic acid maybe it tastes bad. Maybe the mice just aren't eating as much food. Or maybe it's suppressing the appetite, working in the brain and suppressing the appetite. Maybe it's making them nauseated, so they don't want to eat anymore. So that's one way, reduce food intake. The other way you could reduce obesity is to increase calorie burning, okay? So increase your burning of calories. And you could do that a couple of ways. You could increase the basal calorie burning, just what do we burn normally? Or you could increase activity. So anyway, so we investigated these possibilities. So here what we have are data on energy expenditure or calorie burning and then food intake. And we have two conditions, high fat diet and then the high fat diet with ursolic acid. And we have we've broken it into two different periods. So mice, the dark period, the nighttime and daytime. Mice are nocturnal animals, so they're actually more active during the dark period. So that would be like equivalent to our light period. So first we looked at energy expenditure, calorie burning. And as you can see, it's increased throughout the day. Even when the mice are sleeping, they have increased energy expenditure. We also looked at the activity of these mice. We have these cages that have infrared beams, and we can see how many times do the mice break the beams, and that measures how active they are, and that was not changed. So what we're seeing here is an increase in basal energy expenditure, resting energy expenditure, and that's actually consistent with having more muscle. So your muscle mass is the main determinant of your basal energy expenditure. When you have more muscle, you burn more calories, and so we think that this is all secondary to the effects on muscle. Now, we also looked at food intake because, again, another way that you could reduce obesity is by reducing food intake. But as you can see, during the dark period when they're active and really eating, they're actually eating more, but they're gaining less weight because they're burning more calories. Okay, so this is again consistent with an increase in the amount of muscle. We saw a similar thing with tomatidine. So that was all ursolic acid. A similar thing with tomatidine. So here we have weeks on the diet with and without tomatidine. And then we put the animals in a little MRI machine, and we measured the amount of muscle and fat. And as you can see, the amount of muscle or lean mass increases, and the amount of fat decreases. But if you look at the total body weight, there's no difference. The compound doesn't affect total body weight. So you gain the mice are gaining lean mass and losing fat mass. And if we look at the cells under the microscope, we see that the muscle fibers are becoming bigger and the fat cells are becoming smaller. So right now, that's what we know about how these compounds work, and this is our current sort of model. We don't know that this is true, but this is what we think is going on. So we think that ursolic acid and tomatidine, these two chemicals that come from apples and tomatoes, they act on some receptors, some proteins in skeletal muscle fibers. We don't know what these receptors are, and this is our dream of our laboratory is to find these, and that's what we're very much looking for right now. However, these receptors somehow inhibit muscle atrophy pathways, things like ATF4. They also stimulate hypertrophy pathways, things like mTOR. And through those combination of effects, that leads to a change in muscle gene expression and an mRNA expression signature that's roughly opposite to the signature of muscle atrophy. When you do this, when you change gene expression in this way in a muscle, that leads to increased strength, quality, mass, and energy expenditure, and it provides some protection against not just muscle atrophy, but also obesity and the related conditions. So we still have a lot of work to do on this, and we're trying very hard to translate this onto other things. I'll show you one thing that we know so far. We started in mice. Now we got to get into humans. That's a long process. We're beginning to do that with Dr. Rasmussen's group. But we're starting now to see some data from other groups around the world that this is actually turning out to also be potentially helpful in people. So for example, here's a study that came out of Korea recently. They took healthy male athletes. Their average age was 29. They gave them either placebo or ursolic acid. They gave them the amount of ursolic acid they gave was 150 milligrams 3 times a day. And what many people want to know is how many apples is that? It turns out it's 9 apples a day, okay, for 8 weeks. So what they found in these athletes is that if they were on ursolic acid, they got a significant increase in muscle strength. It was between 6 and 12% depending on the muscle group. And they also saw a significant decrease in body fat. There was a 26% decrease. So now we're very, based on these data and the data that we have from mice, we're very excited now to try and begin looking at this in healthy aging. So this slide I'm going to talk about potential areas, ways that we're trying to make use of this information. So first of all, healthy aging. We would like to see these developed, after they're thoroughly tested, developed as food ingredients that could help people maintain their muscle mass over a long period of time. Just something that you could incorporate into food and make help prevent problems with your muscles during aging. We're also using these compounds. We're beginning to work with a really outstanding chemist who has invented several different drugs that are on the market. We're working with him and taking ursolic acid and tomatidine and using it as a way to make medicines. So we're trying to make medicines against muscle weakness and atrophy. We're really hopeful that maybe, based on what we're seeing in mice, that some of these medicines and some of these food ingredients could potentially be helpful towards other things like obesity and type 2 diabetes. So that we're hoping that could be a side benefit of the work on muscle atrophy. And finally, I'll mention we're also thinking about this as pets Pets are important to me, to a lot of people. We have pets, they have similar problems. We think this could be helpful there. And then finally, something we never would have thought of when we started this work is, right now we have a number of feed additives in the agriculture industry, but some of them are not really, we need to get them out of the food supply. Things like antibiotics, we got to get antibiotics out of the food supply. We got to get things like beta agonists out of the food supply. And so we really need a newer generation of safe and effective feed additives. And so we're trying to go down that route and try and help. Hopefully we can do something to help that issue. So with that, I'd just like to thank you all very much for your attention, acknowledge my colleagues at the University of Iowa, also at this company, Emion, that Dr. Rasmussen mentioned. This is a startup company that we started, spun out of the University of Iowa. We set it up as a vehicle to really try and translate some of this work into products that could hopefully benefit people. So thanks again for your attention. If you have any questions, please let me know.
So we have time for 10 minutes of questions or so. One thing I failed to mention is that Dr. Adams sees patients every week. So he's doing great basic science research, but he's also seeing human beings every week and dealing with them. So if you have any practical questions or any basic science questions, the floor is now open for questions. Yes.
Oh, yeah, I'm a VA doctor. We have a VA at Iowa. It's across the street from the med school, and so I have some research grants from the VA, and I see patients at the VA part-time. Part of the university, part of the VA. Yeah.
It's a good organization.
It is a good organization.
Question regarding how you came up with amounts to
Yeah, that's a great question. I didn't go through all that, but when you find something, you have to test a wide range of different doses, and we did that. And what I was kind of showing you is sort of a high level of once we found the right dose. But everything is that's a very important practical aspect of doing this work. Yeah.
So do you recommend, like my mother used to say, apple a day keeps the doctor?
I do.
He should be.
I would. Why not? Yeah. Yeah, I do. Yeah.
So how did you come up with apples and tomatoes?
Well, I'll tell you, we weren't looking for apples and tomatoes. We were just looking for chemicals, and it turned out that we found chemicals that just happened to be there. Now, as you're doing this, when you're trying to find drugs, one thing where it often breaks down is safety, right? So when we were doing this, when we saw things that were in apples, it's like, if that works, that's what we want. So that's sort of the way. I'm not an apple expert. I actually don't even work with apples themselves.
Will this product be available over the counter, or is it prescription, or what?
Oh, I don't. That's a great question. I don't know yet. It's so early. We're still very much like a very early stage company. The idea is that we would hopefully get large companies to take this forward, and I don't know how to make products myself, so we see ourselves as scientists that can help identify things that could potentially then be taken forward by larger companies. Yeah. Yes, sir.
Did you try the Ursolic acid and Tomatidine together for synergistic effect?
Yeah, we've begun to do some work like that. I don't know the answer yet, though, but it's a good question. It's a hard experiment. These are, it took us. The way I told the story, it's like we were developing them in parallel. We actually did about 3 years of work on ursolic acid, and then that served as proof of concept for the discovery method. Then we went back and found tomatidine. That took about 2 or 3 years to develop, and now we need to put them together. But it's, as you know, like when you combine compounds, you love to have like isobolograms and things like that. Our model systems are just not amenable to that. So it's going to take some luck to figure all that out. Yeah, ideally, we'll find the receptors, then that will clarify things quite a bit. Yeah. Yes, ma'am.
The adverse effects to these products?
We haven't seen any. Yeah. Yes.
How 450 milligrams, is that one pill or is it 20?
Well, it depends on, I don't know of the pill yet, but it's equivalent. So one apple has 50 milligrams of ursolic acid. So, yeah.
Would be a large quantity.
Yeah, we're hoping we could, you know, you could eat, you know, we think it's it'd probably be one pill 3 times a day.
Yeah. Is there a particular reason why you did most of your experiments on male mice, on men? And the second question is, are there differences in the muscle weights, quality, and function between men and women? But most of the experiments
It's all male mice, and it's not because we don't think that's an important issue, but it's just there's a lot more variability in female mice. There's an estrus cycle and things like this. So the most efficient way is to study the male mice and then go back and do the female mice, and that's what we're doing now. But it's a separate experiment actually. Are there sex-specific differences and things like that? So I don't know the answer, but it's an important question. I agree. Yes.
And the question with regards to those apples versus the acid itself, which means if you eat 9 apples, would it really give you the same effect with those extracted? With somebody who has something in these apples which will actually prevent absorption, it really will not give the same effect.
Yeah, it's possible.
And the second question is, this was only done in healthy mice, and how this will affect the chronic sick mice, specifically if they have a lot of inflammation.
Yeah, I mean, we've done, well, you may not have seen the beginning of the talk, but we started with starvation models, limb casting models, aging. We've done some experiments with diabetes and obesity. So those are the models. There are a lot of things more that would be interesting, like cancer models, heart failure models. We haven't done that. So yeah. Yes.
I think there's something when your apple compared between the skin of the apple and the pulp.
Yeah.
The skin will feel very better.
That's right. It's in the skin of the apple.
We don't need the pulp.
Don't need to. That's right. There's nothing in the pulp. None of this in the pulp. Yeah. Okay. Yes.
Do you know the turnover of these molecules? Like, do you have to take 450 milligrams daily, or
We
can
don't
it
know.
be 9 ounces a week?
Yeah, it's a good question. We don't know. Yes.
So, everything that you presented talks about atrophy as being a pathology, a problem. I wonder, in your research, especially with the animals, did you discover a function for it? And in the process of doing so, by basically causing it not to happen, was there an adverse effect of that not happening?
Well, I think it's a really interesting question. We don't know the answer. Why do we have atrophy? And I would just emphasize, we never totally blocked atrophy. So we reduced it. Even when, we're blocking it partly. I don't know if you block it all, if that's going to be bad or not. When we started doing these experiments, classically, the reason you undergo muscle atrophy is to liberate amino acids for gluconeogenesis during starvation. And so we thought if we fasted animals that had reduced muscle atrophy, they might become hypoglycemic, possibly moribund. That didn't happen. But maybe it's just because we haven't inhibited all the atrophy. But it's a great question. Why do we have these pathways? Yes.
I think there's been a lot of work showing strength training is beneficial to reduce muscle weakness and atrophy. Have you compared, I don't know how you compare your work to the benefits of strength training?
We haven't done that, and that's really not within our sort of expertise. But After we published our first work on ursolic acid, some groups have begun to look at that. And there's one paper using, it was a well-controlled animal study where they, could electrically stimulate a muscle, do resistance exercise. They did it with and without ursolic acid, and they saw synergistic activation of the mTOR pathway. We know that resistance exercise activates it. They found that if they administered ursolic acid, they got a greater effect. So maybe there's something there.
I have a question. It's very interesting to find the synergism between the muscle hypertrophy and increased strength. There have been a lot of studies actually showing that these two are not regulated by the same manner. And you've actually achieved both. That's my first question. And second, if you look in the cardiac muscle, a lot of anti-TGF-beta molecules that actually been used in the cardiology, they've been also shown to increase muscle strength, but not hypertrophy.
Yeah.
And at least, did you look in the cardiac muscle with these molecules?
Yeah, we've looked at cardiac, size extensively. We've never been able to see any effect on cardiac size. We also can put the mice on these accelerating treadmills and they run farther. So I think the hearts are doing fine. Why does it increase muscle mass and strength and quality? It's the way we found them. When you find things that affect the whole pattern, you shift the whole pattern back the other way. You're not targeting one pathway. And I think that's why things like, for example, myostatin inhibitors, things like this are not going to work, and they're not working. And we know they reduce muscle quality. They increase muscle mass, great, but they don't increase strength. And muscle quality actually declines. It's because it's like a sledgehammer hitting one pathway. But if you do it this way, you hit all the pathways, and you just tweak them all back the other way. And I think that's why we're seeing this. And by the way, the effect is greater on strength than muscle mass because you see the specific force increase. And that's really what we want. Is for patients, I don't think we want to push the cosmetic aspect. I mean, we want the function, not the muscle mass so much.
Do you think it's possible that it's sort of, I say, then kind of stimulant because the mice became more active?
No, they did not become more active.
But they ate more and they moved more.
No, they have a higher resting energy expenditure. Their activity is the same.
I thought you said that the infrared and do you know?
I misspoke. The infrared was unchanged. Yeah.
Oh, okay.
Yeah.
So the activity didn't change.
Correct.
You didn't say increased.
That's good. That's what you see with an increased energy expenditure.
Yeah.
I see more. Actually, and I'll tell you one thing that you can't see from that experiment, but with a high-fat diet, as mice become more obese, their energy expenditure actually declines. Their food intake declines. And this is just preventing that decline. Last question. Yes?
Back to your apple again. Do you have different types of apples?
Okay. I'm not an apple expert, but I have heard that question once or twice. And so I went to the literature. One group surveyed all the different types of apples, and they tested like a bunch of them. And one that had the most ursolic acid was Fuji apples. So, okay. Thank you.
UTMB Health, working together to work wonders.